Hello and welcome to today's webinar, Sample Size for Pilot Studies, Maximizing the Value of Pilot Data. So the hope with today's webinar is to give you an overview of what pilot studies are, how good design and appropriate sample size can ensure that you get the maximum value from your pilot study, and also discuss the idea of internal pilots, which may be a compromise, an adaptive design, which allows you to get all the advantages of a pilot study, but without the waste that's associated with running an external pilot study. So today's webinar is presented by Dotmatics nQuery and is obviously part of our monthly series of webinars. Last month, for example, was on the effect size and how it interacts with power calculations. Before we go any further, just some frequently asked questions. Is this webinar being recorded? Yes, the webinar and the slides will be sent to you after this webinar is complete, either later today or at the latest early tomorrow. In terms of questions, there should be a question button along the side of your webinar panel, a little question mark inside a bubble. If you select that, you can put in your question. If time allows, I will answer some questions at the end live, but even if I don't, I will answer all questions via email after the webinar is complete, and I will likely get a response to those people, even if I do answer their question live today as well. In terms of the discussion of topics today, there is an extensive references section at the end of these slides. So if there's any topic that you want to know more in detail about, those might be good resources to start thinking about that. Okay, so without further ado, as I said, today's webinar, Sample Size of Pilot Studies, the hope here is to give you, a good idea of how big pilot studies tend to be, but more importantly, how you can find a size for your pilot study that's more appropriate for whatever the objective of your trial happens to be. Before any further, my name is Ron Fitzpatrick. I'm the head of statistics here at nQuery. I've been at nQuery over a decade now, since nQuery entered 3.0, and very excited to talk about a feature set that we added in the nQuery 9.3 release last year regarding sample size for pilot studies. Just in terms of the agenda today, we're going to cover pilot studies at a high level, make sure we're all on the same page in terms of what we're talking about today. Then we'll talk about the specific topic of sample size for pilot studies, and then we'll talk about internal pilot studies, which is a type of blinded adaptive design, which perhaps can combine the advantages of a pilot study without the disadvantages of the relative research waste, or not being able to use the pilot data in the final analysis. And at the end, depending on time, we'll have conclusion and discussions. nQuery is your complete solution for optimizing your clinical trial design, covering sample size determination, power calculations, adaptive design, Bayesian sample size for pretty much every stage of the clinical research process, from preclinical research to early phase, phase 1, phase 2 designs, to confirmatory trials, which is the biggest emphasis of the software, especially for adaptive design, and for post-marketing studies. I actually covered post-marketing studies a few months ago in our webinar series if you're interested in phase 4 trials. Just to note, nQuery is a tiered product. What that means is that basically you get a different feature set depending which tier you purchase. And if you purchase a higher tier, you get all the features from the lower tiers. So our basic tier, which is available to all of our customers, is focused on sample size and power calculations for a variety of different scenarios, including hypothesis types, endpoints, etc. Plus is focused in our Bayesian features, which at the moment also includes our pilot study features, which are basically pseudo Bayesian, I would argue, and that's what's in that tier. Pro is focused in on our adaptive design features. That's by far our most popular tier, and it's one which is of great interest if you're in the phase 3 stage, especially for biopharmaceutical development or biotechnology development. And Expert is focused in on our simulation tools for milestone prediction and similar problems. If you're interested in knowing more about each of these tiers and what is in each tier versus the other, there's a URL at the bottom of this slide which you can select when the slides are sent to you later on. I should also note that if you have nQuery and have a lower tier and want to try out the higher level features, you can try for free in your browser nQuery for 14 days. Just you require an email to send you out a link to try it within your browser. And I would also note if you don't have nQuery at all, that's also a good opportunity to try the software and see if it will fulfill your needs for clinical trial design. So pilot studies, what are we talking about here? Well, effectively, a pilot study is a small study typically undertaken prior to some planned major study or clinical trial or similar. And the idea here is that the pilot study will give us additional context, information, and understanding about what we expect to actually happen in that main study, its operating characteristics. Now, in some cases, the pilot study might actually be the basis on which we're going to justify doing that main study, but in some cases, the main study is going to happen anyway, and the pilot study is simply there as a supplementary piece of information to hopefully give us better planning tools, for example, in our sample size determination. So, pilot studies in terms of their objectives and how they're evaluated, etc., is, there's not much commonality across them. And there's a lot of different reasons why you might run a pilot study. And what I would say is that the primary thing that you should be thinking about is the main study. Like, what questions are you trying to answer in that? So I suppose one of the major questions, if your pilot study has been used to justify that main study occurring, is the feasibility. Or assessing the main trial feasibility. So this might be things like trying to get a reasonable estimate for the effect size. This might be like getting a reasonable estimate for the variability, the variance, for example. It might be trying to get a reasonable estimate for how long it's going to take to recruit in the main study, or how much attrition there'll be in the main study. And all of these are kind of fall into maybe potential issues or potential problems that would affect the trial feasibility, that may make that future trial perhaps not likely to be able to achieve the goals that you believe it could set out if the right things came together. You may also want to use a pilot study to try out some new logistical choices or statistical choices or trial design choices. And an obvious case where even if it's not your primary objective, if you're going to do a pilot study, there is often value in using the data that you gathered there to generate updated estimates for certain parameters that might be used in your future sample size calculation, such as the variance if you're doing a two-sample t-test. I would say in terms of like how pilot studies are run and in terms of like design choices, etc., there isn't a lot of standardization for pilot studies. It's effectively a kind of wild west to a certain extent. That is changing. I'll talk about it in a second. But I will generally say is that pilot studies are often done on a pragmatic basis in response to some request in terms of getting a grant application or something similar. And so what often happens is that those practical constraints, those practical concerns often take priority over the design choices and the statistical considerations. But what I would argue in the next slide is that ignoring those design statistical choices means that the value of the pilot study will be considerably less. And I think what you see out there in the literature is that many pilot studies are not able to really answer the questions that they're supposed to answer, and can often lead to research waste, because a poorly designed pilot study that can't answer the question it's supposed to is obviously a problem in itself, because you're recruiting real subjects, you're spending real time, you're assigning real hours for experts or students, etc. And obviously if the pilot study is not able to answer the question it's supposed to, all that resources effectively wasted. But what I would say even more perniciously is that a poorly designed pilot study can often, bias your actual insights when thinking about what's going to happen in the main trial or when you're planning the main trial. So for example, in the context of a sample size determination for the main trial, if you use the pilot study data, there's a natural inclination to, think about things in a positive manner. There's a certain amount of motivated reasoning. If you're a researcher, you obviously want your intervention, your drug, your therapeutic to work. And so what often happens in practice is that if you run a pilot study, which is say way too small or just poorly designed, if the estimate for say the effect size is much higher than expected initially, you might be a little bit skeptical, but you might go, this is a really good sign for my study, and maybe we can like reduce, increase the expected effect size, and that means the overall sample size required in the main study is reduced a little bit for the same power as if the effect size is a little bit lower. But if I had the inverse, where I had a very small pilot size, but it came out with a very small effect size, the natural inclination I would say for many researchers to go, well, small sample size, not that reliable. We'll just go what we originally thought the effect size estimate would be before we did the pilot study. And basically, you can see there's a pernicious bias there where pilot studies can be used as a justification to underpower your main study, and then that can lead to much larger research waste, because of course those major studies will likely have hundreds, if not thousands, of subjects in them compared to what feels like a small pilot size. So obviously a poorly conducted pilot study intrinsically has a cost, because that that pilot study data effectively isn't of much value, but also the kind of poor decisions that can emanate from trusting a poorly designed pilot study can then have major implications for your future study as well. And so I think all that comes together to say that in recent years, there's been significant work done to improve the reporting guidelines and the design guidelines for pilot studies. And so I'd like to particularly point to the Consort extension for 2016, where they talked about some of the reporting aspects of your pilot study. And while this is mostly in reference to like if you're going to write this up in a grant proposal or if you're going to write this up in the literature, I think these checklist items are very good for at least getting a sense of the kind of things you should be thinking about from a design perspective, ensure that your pilot study is able to answer the questions that you want and is going to be able to do them in a robust, precise way. And so you can see this has things like think about your background and objectives, think about your trial design, who you're going to let into the trial, what outcomes you're going to measure, how large is it going to be, how are you going to deal with randomization, etc. And so what I've done here is kind of given a kind of Cliff Notes version of the consort statement and some of the other resources that are available in the references that talk about good design for pilot studies. And what I would say is here that start with the top. These are the most important. But by the time we get to statistical issues, many of the answers in this last section should be obvious or should naturally flow from the decisions or choices you've made at the upper levels. So in terms of study objectives, the first thing you should be thinking about is, why am I running a pilot study? And of course, the first reason and the most likely reason is that you want to assess the feasibility of your future main trial. So you want to get a reasonable estimate for the effect size. You want to know, is the variability going to be way too high that I'm going to be underpowered given the amount of budget I expect to have for my main study? Is dropout going to be a problem? Do I want to get an estimate for that? And I think the main thing here is that that there's so many different ways you can think about feasibility, and so you want to have a good idea of which of those you think could be an issue, and therefore which ones you're going to prioritize in terms of measuring them. And that will then affect some of the constraints and choices you'll make later on. And I think if you're going to get parameter estimates, and you're almost certainly going to be measuring at least one or two things, whether that be the effect size, the variance, or the dropout, you also hopefully want to bound your degree of uncertainty. So, your pilot study should hopefully give you a reasonable range that you would expect the actual value to fall in your main study. And obviously we can use statistical theory like confidence intervals or credible intervals to hopefully give you a reasonable estimate of that uncertainty based on the size of the pilot study. If you're going to be doing experimentation, if you're going to be trying out new design tools or logistics or whatever, you obviously should be thinking about, what kind of problems should you expect to have? You should have some background idea of like what's the kind of weaknesses of this method, or what's the areas where this might fall down. And hopefully the pilot study can be optimized to maximize your chance of finding that problem if it does exist. And that thinking about that beforehand is useful rather than just hoping the pilot study just turns some stuff up. Now, some stuff's going to be found. There is a certain amount of discovery stage to doing a pilot study, but you should be thinking and hopefully predicting some of the more important ones a priori. But then I think you want to know what you want to answer. So that's really important. You should know that upfront. Only then should you think about, in my opinion, the constraints. And obviously the constraints are going to be really important. And the reality is there's going to be a tension between the kind of two objectives that you have for a pilot study. You want the pilot study to be realistic, to be actually doable within the constraints of your actual research group or the company that you're within, but you also want it to be sufficiently high quality that's going to actually add value when we get onto the actual design and discussing what's going to happen in our main trial, whether that be trying to justify doing the main trial via grant proposal, or whether that be doing your sample size justification for your main trial. Those are the kind of you're balancing those two, and there's a certain tension because we're doing a pilot study not to find out directly about what we're really interested in, otherwise we'd be doing the main study straight up. We're trying to hopefully get additional context that will make that main study better. But you obviously don't want to waste too much resources on the exploration stage, in case that would mean that we take resources away that could be used for the main study. So in terms of resource, the obvious things here are the budget. There's obviously access to expertise, like statistical expertise, like clinical expertise, etc. And I think one obvious thing to do is to think about ways that you can share resources between the external pilot and your main study, whether that be via statistical mechanisms like Bayesian methods, like a power prior. But I suppose I'd say more practically, you're usually talking here about things like, if you're recruiting people for a pilot study who are from your target population, perhaps that could be a short list of people who could then immediately be a part of the queue to be recruited into your main study, assuming, of course, that they still have the condition in a suitable area. To then quicken the initial part of your recruitment process for the main trial. Think about the synergies that can exist between your main trial and your pilot trial in terms of experts, in terms of things like that. And then time is obviously going to be a major consideration. You don't want this pilot study to run forever. You want it to happen within a reasonable time frame. You want to know ideally roughly what that time frame is going to be, like the deadline for when you need to kind of cut this down and kind of get the results out there so they can actually feed into the processes for justifying or designing your future study. That can have a major effect on your endpoint. Like you want to think about what kind of endpoints can we do. Some of them are going to be variable. So if you were doing, it's very rare you do this in a pilot study, but if you had a time to event endpoint or even a count endpoint, that has more of a variable follow-up time than measuring a mean after a fixed period. So there's definitely something to keep in mind. And then recruitment, you obviously want to think how quickly can I get people in? How quickly can I get people in stuff? And obviously that might influence like who you want to include, because having very restrictive criteria for who gets into the study will slow down your recruitment on average than perhaps having more liberal criteria. But then that kind of directly comes into the third point here of comparability. Like the more similar our pilot study is to the main study, the more portable or more accurate or more confidence we can have that the estimates that we take from that pilot study will actually be what will manifest in the main study. So we want the pilot study to be sufficiently similar to our main study, such that the inferences that we make from the pilot study are actually appropriate when we just are making decisions about what's going to happen in our main study. Like, for example, in the sample size determination, for example, when talking about, like, well, is the feasibility actually true? And so there's a kind of a tension there, because of course the main study, we're probably going to do a randomized controlled trial. We're probably going to have quite restrictive inclusion exclusion criteria. There's going to be a lot of logistical work to kind of make that work, like going to hospitals and recruiting there, whereas a pilot study might be 20, 30, 40 people, and that might be done on a more ad hoc basis. But we don't want those two to diverge too much in terms of their design, in terms of statistical analysis, because then how much value will the pilot study add when we try and get a good idea of what's going to happen in that main study. And so that directly feeds into the study design. So the endpoint selection, like, do we want to use the same endpoint as in the future main study? That's a natural thing to do, but perhaps the endpoint in the future study is a gold standard endpoint, like overall survival. So perhaps we need to rely on a surrogate endpoint, like response rate. And of course, the surrogate, we then need to think about how well it is able to predict the primary endpoint of interest. And we may also want to think, like, if we have lots of endpoints, if we were doing some kind of testing, like, do we need to do adjustments for that? And of course, when we're sizing our pilot size, are we only thinking about one endpoint, or we need to think about all the other ones, and how do we kind of reach a consensus or a compromise between all of those different endpoints we might measure? But I think that choice of endpoint, I think it's very often going to be the case that you're going to require a surrogate compared to what might actually happen in a main trial, especially if the main trial is like a pivotal trial of some type. And like, that's not uncommon within the clinical trial process itself. Like, a lot of the methods you'll see for pilot studies are actually quite similar or inherited from phase 2 designs, where in phase 2 trials, you're often using an overall response rate as, say, a surrogate for oncology trials, but the main pivotal trials, the phase 3 trials, are based on overall survival or progression-free survival. And there's a whole debate right now within the regulatory agencies and stuff about, which surrogates work, which surrogates have value. But if you're doing a pilot study, that is something you should definitely be cognizant of. But of course, you're picking a surrogate, presumably because it's more efficient in terms of time. You can get a surrogate endpoint on a reliable basis at a quicker time, with perhaps less intervention, like less, logistical problems of following up people, whether they died enough. This is just a simple measurement when people come to you in the clinic, and that could all, affect what you want to do in terms of your endpoint selection. In terms of number of arms, I would say, most pilot studies are single arm. It's the most common. It's obviously the easiest to run, like a cohort design. But I would say, don't dismiss the idea of doing randomization. Even if you only have like 40 people, randomizing to 20 and 20, that adds a lot of value, especially if you're trying to get an effect size estimate. So if you're trying to get an estimate of the mean difference or the proportion difference or the odds ratio, hazard ratio, etc., I think it's definitely a case that, you could definitely just take the mean from the one group, compare that to like the baseline or the historical data, and get a, inference about the estimate there. But what I would say is that if you do randomization, the portability of the estimate is likely to be improved compared to the single arm trial. And in terms of subject selection, this kind of very much directly goes into that comparability point where, if you're using only the target population is going to be used in the main trial, like, that's obviously better in terms of the portability of your estimate, almost certainly. But obviously, if you have restrictive inclusion exclusion criteria, if you have certain mechanism, like you want to get a geographically wide set of sample in the actual main study, those all might be things which just aren't realistic for your pilot trial. But you do want to consider, maybe, when you take the pilot study data to and inferences, to be skeptical. Just the further away your target population in your main trial will be from what you did in your pilot study, that will affect and can induce biases, and certainly you should think about what biases could occur and appropriate adjustments that may be required there. And so that all kind of feeds into the statistical issues. Like, obviously, if you pick an endpoint, you then also need to think a parameter that you're going to use to estimate that endpoint, whether that be, the standard deviation for variability, whether that be the mean difference, etc. And, if you want to consider some scales transformations to kind of maximize your small sample size that may also be considered. Statistical methods, if you're talking about bounding the parameter estimate, you want to probably use a statistical interval, and then you have a choice between like confidence intervals or prediction intervals or credible Bayesian intervals. And you want to think about what your success criterion is, and your success criterion might be the upper bound for a confidence interval at 80%. And so that's one of the things I'll talk about later on. And then, of course, sample size determination. How big will the pilot study be? And obviously, that's kind of feeding into the fact you're making a compromise between, you want to know as much as you can, but obviously the resources are quite constrained. So how do we balance these two issues? And these are usually based on a rule of thumb or sample size formula, or on practical constraints, or some combination thereof. So in terms of pilot study sample size, your pilot sample size should ideally be based on what is your primary objectives of interest. So if you have a safety endpoint, you should ideally, be finding like a confidence interval for the safety endpoint, or a confidence interval for dropout or something else if you're worried about practical issues. The trial estimates you're getting are really feeding into the feasibility assessment, whether that be for the effect size or the variance, etc. And so pilot sample sizes, if you read the literature, I would say the majority of the ones you're going to see are commonly based on what I call rules of thumb or heuristics, or realistically, they also take into account practical constraints. I would say most of the rules of thumb are really for continuous endpoints and are most related to uncertainty around the variance. So the variance being higher than expected. And that's basically true for most of the inputs in this table on the left, or sorry, on the right, which are for the two-sample t-test. So the main study is going to be a two-sample t-test. The pilot study may or may not be a two-arm trial, or it could be a one-arm trial. But basically, this is giving the total sample sizes you might require to get a reasonable estimate from your pilot study for the variance, such that you can then put that in your sample size determination formula for the main study and be reasonably confident that you're not going to underpower your trial. What I would say is that if you're going to just use practical basis to pick your pilot size, like we have budget for 20 people, that's all we can do. Be explicit about that. There's a good summary paper by Lakins on sample size determination, it's a primer, just generally. But I think the point is well made there, that if you're basing it on practical stuff, say it up front. And that doesn't mean you don't have to worry about sample size at all. You can, given a fixed sample size, investigate what would the implications be that for the precision of your estimate from the pilot study. And I'll talk about that during the demo aspect. What I would also note here is that, as I said, these rules of thumb, and they're cited very widely, are all pretty much for a continuous endpoint. But if you were to have like a binary endpoint, like a proportion, then you note, and it actually comes up in this tier paper here, it's a really good simulation study for pilot size. You're basically almost doubling your pilot size required for a reasonable estimate for a nuisance parameter, in that case, the two proportions or the overall proportion. And what I would say, therefore, is like, it's a point I made in a webinar I did earlier this year about dichotomania, the tendency of people to turn continuous endpoints into binary endpoints. You lose a lot of efficiency doing that. And in a pilot study where the sample size is very limited, obviously the cost of the inefficiency is magnified compared to a trial with thousands of people. But even in every trial, I would strongly recommend that if you have a continuous endpoint, not to turn it into a binary one. And I think doubling your pilot size is a very obvious way to communicate that to your collaborators, that let's stick with the continuous endpoint. Let's even if it's a slightly more complicated analysis to run in their opinion. And like, most of these rules of thumb, thankfully, are based on calculations that can be run for a specific trial. As I said, for a two-sample t-test as the main trial, and you're running a one-arm pilot size or a two-arm pilot size pilot. And, you can use these methods to explore if your assumptions are reasonable. You'd hopefully get an idea about, whether your trial actually is feasible or would make sense to do in the first place. Like, and if you can't reach like a minimum of 20, this is basically saying that, for a continuous endpoint, the value from that pilot is likely to be minimal, and you're probably better off not even doing the pilot study. And in that case, perhaps you might consider the internal pilot approach that I mentioned later on. And as I said, even if you pick a sample size based on practical constraints, or you just pick a value from this table on the right, you can still investigate the implications that it has for your parameters of interest. So for example, we're going to show during the demo, you could calculate, for this sample size, what the upper estimate for the variance actually would be, or the standard deviation actually would be. So there's two main methods for doing pilot study sample size determination, the UCL method and the NCT method. Now there's other approaches out there. There's Bayesian approaches, some which just replace the confidence interval in the UCL approach with a credible interval, some which have a decision theoretic framework. It's very interesting, but well beyond the scope of today. And there's also this paper by Wiechbauer, SSE for problem detection, where effectively you want to know the size of pilot you need to be able to find some quote unquote problem. The problem is left undefined. It could be like dropout probability or something like that. You know, you want to find at least one of those incidences, like how much size do I need to be able to have that a reasonable possibility of finding that given some assumed underlying rate. I'll show that very briefly during the demo stage. But in terms of these two methods, UCL and NCT, the UCL method, effectively what we want to do is we want to calculate a confidence interval. In this case, for the papers we're talking about here, the Whitehead paper in particular, you know, for the variance or the standard deviation. The standard deviation obviously just be the square root of the variance in this scenario. And basically, we want to calculate the confidence interval, which is based on the chi-square distribution, basically the ratio of the sample variance to the true variance. Can be related via the chi-square distribution if you include the degrees of freedom, which are related to sample size. And in effect, you know, what we're going to do is we're going to take the upper bound for that confidence interval. And in this case, we're talking about one-sided intervals, but you could just take a two-sided interval at the, you know, half the appropriate level. Or sorry, double the appropriate level, and then you can take it, do it that way. So, for example, you know, if we're using a 95% two-sided interval, that's equivalent to a 0.975, 97.5% one-sided interval. But we'll talk about one-sided intervals. I think you should probably just do it that way because we're not worried about the lower, like, you know, the variability being much lower than expected. We're not really too concerned about that because we want to be pessimistic, right? Because, you know, the pilot study is quite small and we're just trying to find an upper bound, like a pessimistic assumption about what might happen. So we have a reasonable chance that the variance will be at that value or lower in our actual main study. And that ensures that if, say, for example, we're using this in a sample size calculation for the main study, that we're not going to be underpowered in that main study. And so basically we're talking here, you calculate just as you would calculate confidence interval for any sample or any study, you know, for the variance. And basically we're taking that upper bound and then we're inputting it directly into our sample size calculation. And now that's, you know, the sample size we're going to put into our main study based on, you know, some fixed effect size that we're interested in. And what I would say is that, like, I'm not going to talk about today, but this is, you know, pretty extensible to pretty much any other parameter that you could put into a future sample size calculation. It could be the effect size, it could be the dispersion parameter, you know, it could be the overall the two proportions in a chi-squared test. But what I would say is, of course, you know, keep an eye on whether you're interested in quote unquote the upper bound or the lower bound. So obviously that depends on the relationship between that variable and the power. So, you know, the higher the variance is, the lower the power is. So we want to take the upper bound, but obviously the lower the effect size is, the lower the power. So if we were doing a constant interval for the effect size, like say the mean difference, we'd usually be looking for the lower bound of the interval, or a lower, you know, a constant interval that goes in the lower direction, you know, negative values that direction. And then the NCT method, it's a bit more complex, but effectively the argument from Julius, the paper that is originally from, although it's discussed in this Whitehead paper, I'll talk about it in a second. But in effect, the argument from Julius is that there's an intrinsic uncertainty around a sample standard deviation. So if you take a sample standard deviation, we know that that has it's basically a random variable. You consider that to be, you know, whatever your sample variance to be, a pull from the random variable, and that's actually quite closely related to the distribution constant interval stuff up here for the S squared. And in effect, what he argues that, well, rather than just taking, you know, naively taking the sample standard deviation and inputting that directly into our power calculation without adjustment, just assume it's the population variance, the population standard deviation. He says, why not take the expected power from the assumption about the S squared? So in effect, you know, if we consider the S, that whatever our sample standard deviation for the pilot study is a draw from a random distribution, if we average over, given whatever that sample happens to be, you know, what the implied random distribution would be for the S squared, we can then get the expected or average power over that distribution, and then that gives us a slightly adjusted power formula given here to get the sample size such that we've adjusted slightly upwards for the fact that we had that uncertainty in our estimate of the variance, because of course, you know, that involved random sampling. And what I would say is that the adjustment for the UCL will tend to be a fair amount higher than for the NCT method, because the NCT method is really just, you know, it's a really small adjustment. It's just like, well, look, we think your standard deviation is a good estimate because you obviously sampled it presumably appropriately, but, you know, we're just taking into account the fact that, you know, because it's a random pull from the distribution, we'll take the expected basically the power function averaged over that that implied distribution rather than just taking the naive point estimate directly. And what I would say is that these can be done separately. So you can do these two methods and kind of just calculate the sample size straight off from that. And that's what these formulas do. But there was also an extension that's discussed in this Whitehead paper from 2016, where they talk about a mechanism via which you can minimize the overall program sample size. So rather than just going, here's what the sample size will be based on a pilot study that we picked from the air, or we picked based on this table over here, but rather what if we wanted to minimize the total sample size across both our external pilot where that data will not be used in the main analysis and our actual main study where we actually obviously that data will end up being used in our final hypothesis test of interest. And so basically this method provides a mechanism via which you can basically reduce the overall sample size to its minimum if we include the total of the program. One thing to note here is that the UCL approach probably easier to explain. It also has less things that affect it. It's basically just affected by your level of confidence, the level of confidence you have, and the size of the pilot study. Whereas the NCT approach not only relies on those two, but also is dependent on the final alpha and the final power in your main analysis. So however the levels that you're going to test and power for in your final analysis, that will also have an influence on the size of pilot study in the context of the NCT approach. And that's actually where this Whitehead, this much wider range of Whitehead is basically taking into account that those differences in effect sizes and the fact that do you get different results if you're powering for 80 or 90%. Whereas for the UCL approach, that wouldn't have a direct effect, at least if you're just doing these formulas. That would have effect if you're doing the minimizing the total sample size for the program. That would primarily just be on the end of changing the main study sample size rather than the pilot size. So basically what I've taken here is an excerpt from the book from McKean, the sample size for clinical laboratory and epidemiology studies. And this basically covers pilot study sample size in more detail if you're interested in knowing more. Julius actually himself came out with a second edition of his sample size book. Both of them are included in the references. So if you want to know more, both those books are really good resources and can become highly recommended. But basically, in this case, they were planning a main study with 0.05 significance level at two-sided level and a power of 90%. They were going to assume that the standardized effect size, the Cohen's effect size is 0.2, and that's just equal to the mean difference divided by the standard deviation. Basically, they said for UCL 80%, UCL 95%, and NCT approaches, these were the pilot sizes: 95, 104, and 56. Of course, we see there already, a more liberal confidence interval, like one with a lower level of confidence level, will require typically lower pilot sizes, and the NCT will be lower than any reasonable UCL. Like you're probably not going to, doing any UCL less than 50% would actually probably be harmful in this case. And you can see, therefore, that has a direct effect where the sample sizes, unsurprisingly, will tend to be highly affected by the relation between these three. The higher the pilot size is, the higher the total program size is, effectively. And so what we're going to do is we'll go into nQuery and we'll basically replicate those results from the McKean book. So basically, we'll do in the first column the 80% UCL approach, in the second column the 95% UCL, and in the third column we will do the non-central T approach. So like the first set of these parameters are all going to be the same across our two. So we're going to make the simplifying assumption that the mean difference that we're expecting is 0.2, and that the standard deviation, and therefore the variance, since the square root of 1 is 1, is equal to 1. And so the sample size allocation ratio in the main study is 1, but assume here, actually, this table is assuming a one-arm pilot study. So we're only going to have one arm in the pilot study, which we're going to use to re-estimate the variance effectively compared to what our initial estimate might have been. And so I'll just copy and paste these, and we're obviously going to have 90% power in our main study, and we can kind of just copy and paste these across into columns 2 and 3. So if you're not familiar with nQuery, in effect, each row corresponds to either an input or an output required for the calculation. And basically, you can get some context for what each row is by looking at the help card here on the right, which gives a brief overview of what this input is. The yellow rows are ones which we can calculate for. Those are kind of our solver rows, as we call them internally. And the white rows are what we call input rows, things that are required to be inputted so we can do the calculation. And so we can see here that we put in this column 0.8, and then we put 0.95 in this column. This corresponds to an 80% confidence interval, one-sided confidence interval, and a 95% one-sided confidence interval. And then we just put in our pilot standard deviation of 1 into each one of these, and you can see that we now replicate the same sample sizes as from the McKean paper. Now there's a difference of 1 for some of these, but that's just due to the fact that for equal sample size, McKean will round up so that the value is divisible by 2, effectively. But for practical purposes, this should replicate what we originally had in the slide here. And obviously you can see here, the general point that, the more you increase your level of confidence, the more restrictive you want your interval to be, the more it's going to push your estimate for the standard deviation, the upper limit of the interval, further in the upwards direction on average. Even though we have a higher sample size, the upper bound for this estimate actually will still be higher than this estimate, because this is a less restrictive alpha, so that compensates for the lower pilot sample size here. If you're interested in exploring the results more, there's obviously an output statement here, which you could easily copy into that consort statement if you wanted to, for the sample size justification, and basically go from there. And of course, we did it like, in the Whitehead approach, where we're trying to minimize the total sample size across both our pilot study and our main study. And I should mention, there is a bit of a caveat there. We're assuming that pilot samples are equally expensive or equally weighted as main study subjects. That might be a non-realistic assumption. So you could probably, it wouldn't be too difficult to extend to account for that if you want a different weighting, but just something to mention in terms of how this, like, overall sample size is minimized. So we're trying to minimize this row, the total sample size, which is the sum of the pilot sample size and the main study sample size. But let's assume, we're going to cheat here a little bit, because I basically recreated the first 2 columns for the UCL approach, where I've calculated, like, let's assume we had a pilot sample size of 91 and 144, and I just happened to pick these confidence levels of 0.8 and 0.95 for one-sided interval. And basically you could see that based on this, basically the upper limit for the standard deviation in the 0.8 case was 1.07, and for the 95% case, it was 1.108. And what we can do is we can show that, like, let's imagine these pilot studies had just come out of there, we weren't worrying about, like, minimizing the whole thing. We hadn't got these Whitehead, we'd actually just, chosen these arbitrarily, or we'd chosen them based on rules of thumb or something like that. We can go into the 2-sample t-test table here, MTT0. So if we just enter, these values into the standard deviation instead of 1, which is the value we got from our pilot study. So remember, we got what we got from the pilot study was a standard deviation of 1. But, if we want to be pessimistic and kind of want to ensure that there's a high chance that the value in our actual trial would be lower than some upper bound, and then these are the values we've calculated for the upper confidence interval for that. So if we input that instead and then put in our power of 90%, we'll get 603. And if we double that, we get 1206, which is effectively the same as 1205. And of course, if we were to do the same for 1293, we would get the same type of thing. I should also note here, of course, if we had just assumed that, the standard deviation was exactly 1, then, the sample size would be significantly lower. But the worry here is that, because we're, the standard deviation we get from our pilot study is a random sample, this could end up being, an underestimate of the true thing. So we're kind of hedging here by using the upper estimate, such that we now have a much higher degree of confidence that when we power our main study, it's going to actually be appropriately powered. Cool. And if you wanted to think about, for example, the confidence interval for 2 means, and there's a good paper by Julius which talks about, you could think about the efficiency of adding each individual subject on the increasing the precision of your estimate. But you can see here that, for example, with a common standard deviation of 1.07 or 1, we're thinking like the distance from mean to limit for the mean difference, that's all 1.33 or 1.2. So you're talking here about, like, if we assume our mean difference is actually 0.2, we're getting all the way down to, mean differences could be plausibly all the way down to 0.07, and that might feed into your feasibility assessment for your actual main study. Do we have too much probability of getting effect sizes which are, would be problematic for the clinical meaningful results? And then just to briefly mention the Weightbauer method, where basically, we assess, each subject has a 10% chance of having the problem, let's say dropping out or getting some reason to drop out. If we want a 90% chance of finding out that someone will drop out due to some X condition, we need a pilot sample size of 22. And that's kind of summarizing this statement down here. Great. So hopefully that's a reasonably good idea now of how you can do pilot sample size. And we're obviously talking there about external pilots. So this is where you're going to run a pilot study prior to the main study, and you're using this to, understand, get lots of estimates and stuff like that. But in this specific case we talked about today, appropriately sizing the pilot study so that it provides meaningful, useful information that you can calculate the sample size for your main study and be reasonably confident that you will be powering that to the appropriate level based on your uncertainty around the estimate for the variance in the context of a two-sample t-test. But that could also be extended to a number of other parameters. And it's actually easier to talk about those other parameters in the context of the internal pilot, because an external pilot effectively requires you to throw away your pilot data, right? You do your pilot study, you get the insights, but those subjects, their data doesn't end up in the final analysis. They more or less get thrown away. They may, via some Bayesian feature or some kind of pooling mechanism, you could in theory include them, but that's very rare. If you're doing an external pilot, that would go against the idea of doing an external pilot. But what if there was a better way? Well, one better way would be, why not kind of bring the idea of a pilot study, but why not just consider the first portion of your actual main study to be the pilot? And this is what we call an internal pilot study. And this is basically an adaptive design where we use an initial cohort to improve our understanding of some parameter or parameters within our trial, and use that improved understanding to then make changes to the trial while it's still ongoing. For example, increasing the sample size. And so the internal pilot concept is very closely associated with the idea of a blinded adaptive design or a non-comparative adaptive design. And that's not because an internal pilot can't be unblinded. They certainly can be. And much of the initial literature, if you look up internal pilot and the ones that are included in references at the end, like Burkett and Day, those were for an unblinded internal pilot. What was found there typically is that unblinded internal pilots tended to lead to type 1 error inflation, for example, if the basis of a sample size re-estimation. And in effect, if you're going to do an unblinded analysis, where you're going to know so obviously the difference there being, for blinded, you don't know the labels. You get all the subjects' results, but you don't know who is in the treatment and control group. Unblinded means that you do know who's in the treatment control group. But in the unblinded case, the obvious question you would have is, if we know the labels, if we know who's in each group, we can re-estimate the effect size. But at that point, why don't we just do a group sequential design? Why don't we do a sequential design where not only do we make a decision of, say, perhaps do enrichment or increase the sample size, something like that? Why wouldn't the first step be, let's stop the trial early if the evidence is very strongly for or against our treatment being effective? And so that's why effectively I would say unblinded internal pilots have mostly gone out of fashion, and I would say be superseded by group sequential designs and variants thereof, such as promising some designs which are for unblinded sample size re-estimation. And I would say the regulatory perspective on non-comparative, unblinded adaptive designs is very positive. And so you can see here a excerpt, obviously edited down for space, from the FDA 2019 guidance on adaptive design, where they say adaptations based on non-comparative data have no or limited effect on the type 1 error. This makes them an attractive choice, particularly when uncertainty is high. So obviously the uncertainty we might have here is about the variance, which is basically what we talked about in the previous section, or it could be about some other nuisance parameter like the dispersion in the context of a count model. And so, this all suggests that if you're going to do an adaptive design, this internal pilot approach, and it's blinded, there's a high chance that the regulator will be, inclined to view this in a positive light, assuming, of course, that you show the evidence that you'll be able to maintain the blind and that there won't be any funny business. And I would say the most common type of internal pilot design, therefore, is what we call blinded sample size re-estimation, where we take the initial cohort of subjects, we get we re-estimate some value for a nuisance parameter, so something that's not the effect size, it's not the mean difference, it's something like the variance, and then we increase our sample size if our initial estimate was over optimistic. So we thought the variance was going to be lower than actually ended up being, and based on our re-estimate from the internal pilot cohort. And so I provide here just a summary slide on blinded sample size re-estimation. We have webinars in the past on sample size re-estimation in total. But basically, the idea is very simple. Take, say, the first 25% of your subjects from your initially planned main study sample size. So, you did a sample size calculation. It required 100 people per arm. Take the first 25 in each arm, the first 50 people in total, recalculate the variance or recalculate some nuisance parameter, and now re-estimate your sample sizes if that was your initial input into the sample size formula. And the great thing about this is that Friday and Kaiser and a bunch of other papers show that for a wide variety of scenarios that you can basically do that recalculation based on blinded data, based on non-comparative data. Note the non-comparative language is mostly there in reference to being applicable to open label studies, but blinded is perfectly adequate. It's used interchangeably in the adaptive guidance from the FDA, for example. But in effect, you can take your internal pilot cohort where you got a better estimate, you increased your sample size if you were over optimistic about the variance or some other nuisance parameter, and then at the end of the study, whether or not you increase the sample size, you take the internal pilot data and you take the sample after the internal pilot, which, as I said, may or may not be bigger than the initially planned size, and you can treat that as if that was the original plan, as if all those subjects were always planned to be in that study, and just do a standard t-test analysis, standard chi-squared test, standard log rank test, etc. Now, you may want to verify that via simulation. I would know that Friday and Kaiser point out in a separate paper that for non-inferiority, or NI here, our equivalence hypotheses, that type 1 error inflation is a little bit more common, but even there, we're talking about increases from for 0.025 going up to maybe 0.03. So these inflations are not huge, but if you're doing, in the case of not doing, if you're doing non-inferiority or equivalence, maybe a little bit of simulation wouldn't be the worst to verify the type 1 error over a variety of different internal pilot sizes, at different values for the effect size, the assumed effect size, and different values for the nuisance parameter. And I would note that if you are worried about type 1 error control, you can always do a combination test like the inverse normal test, and that will basically guarantee type 1 error control. The main reason you wouldn't want to do that is because those tests rely on using weights based on the original sample size. So, if the internal pilot is like 20% of total sample size, then it will have a weight of 0.2. And then regardless of whether the post-internal pilot cohort is larger than initially planned or is at its original level, that second cohort will only have a weight of 0.8. So obviously, if you increase the sample size, if you double the sample size, if in the unweighted analysis, then that weight will be much higher for that post sample size cohort, because now it's much bigger than originally expected. But in the combination test, it would still be tied in, weighted as if the original sample size for the post internal pilot was still being used. And that, in some arguments, the arguments of some means that if you do a sample size increase, then some of the subjects are affected. The subjects in the post internal pilot cohort are effectively being underweighted. They're being weighted lower than they should be compared to the unadjusted analysis. And what I'd say in terms of the blinded SSR methods, there's kind of two main branches. There's blinded SSR in the case of independent variables, variables that are independent of the effect size, and there's ones in which they are, like the nuisance parameter is dependent on the effect size, or at least using that nuisance parameter to get estimates for the parameters of interest requires making an assumption about the effect size. So for nuisance parameters, which are independent, think about the variance in the case of the two-sample t-test. We talked about that already in the context of external pilots. Over dispersion in the context of count models. For non-independent ones, think for the chi-square distribution, you can calculate the overall proportion, and you can then recalculate the assumed P1, P2 values into the proportions in each group. But you do require to do that recalculation a strong assumption about what you expect the true proportion difference or true odds ratio to be, to do this for the chi-square test for the two-sample case, for example. And if you're interested in knowing further about how do you choose what type of sample size reestimation to do, or what kind of processes you should go through when doing this in practical terms, I can really highly recommend the Pandas resource. The URL is given here in the bottom right. This is a really good resource, not just for sample size reestimation, but for a wide variety of commonly used adaptive designs. At the University of Sheffield, it's a really great group there who are bringing all this information together. Great references, great resources, and a really practically focused guide to what things you should be thinking about for doing an adaptive design. Obviously not the focus of today, but something I would definitely want to shout out, and these excerpts from that are greatly appreciated. So depending on your design, depending on your endpoint, you're going to have different strategies for your blinded SSR. And I have a very brief summary here for kind of four different, two independent group analyses. So the two means things are very similar to the stuff we've already discussed. We're interested in, getting a better estimate for the variance, for the standard deviation, the within group one, of course. And this could be applicable for a two-sample t-test, a two-sample z-test, a man-weighted u-test if you were willing to make some strong assumptions. And in effect, the strategy is exactly the same as what we had for the external pilot. Basically, we're going to, take our internal pilot, we're going to recalculate the variance, input that into our sample size formula, and then adjust our sample size upwards if required, if we were too optimistic about the value of the variance, we were too low in our initial estimate, in our initial sample size calculation. There's a variety of different methods for recalculating the sample size. One that works surprisingly well is just take the blinded cohort, calculate the overall variance, and treat that as if that's the within group variance. And the reason that works is that the overall variance has two components. It has the within group variance and the between group variance, but the between group variance component of the overall variance is very, very small. So you can kind of think if you have two groups, which there's a difference between them, it's technically like a bimodal distribution. But what you find in effect, even if you combine them into a blinded cohort, is that that effect is typically in most clinical trials for most meaningful effect sizes trivial. And so in effect, you could just use the overall variance, input in the sample size formula as if that was the within group variance, and it worked really, really well. And indeed, Friday and Kaiser argued that the fact that it's a little bit higher than it should be, because it obviously the overall variance includes that between group variance component, is actually a good thing, because that offers a little bit of a hedge against the type 1 error control that intrinsically comes from doing a little bit of sample size re-estimation. So from their perspective, it actually, those two things balance out on an ad hoc basis. There's no like formal proof of that. It's just an ad hoc thing to say that just being a little bit pessimistic by adding, keeping the between variance thing in, it actually does no harm. But if you wanted to get rid of the between variance component, there is a proposal from Zucker where you can, if you're willing to make a strong assumption about the true mean difference, you can subtract that small amount based on that assumption and get a slightly downward adjusted variance if you want. There's the upper CI variance estimate. Obviously, at this point, we should be familiar with that. That's basically exactly the same strategy as we just showed for the upper UCL approach for an external pilot. And there is this EM algorithm. I won't go talk about this too much, but it's basically it's an expectation maximization algorithm supposed to kind of that bimodal problem, kind of try to pull those apart based on the data and kind of recreate what the two independent distributions are. But I would say that the literature is very, very negative about this, both from Friday and Kaiser and from a subsequent paper by Axman. But I'm sure Gould and Shi would disagree. Two proportions, in this case, and two survival, these basically work the same. We're going to calculate an overall proportion, or we're going to calculate an overall hazard rate. And basically what we're going to do is we're going to recalculate the per group proportions or per group hazard rates by basically making a strong assumption that the effect size, to say the odds ratio, proportion difference in the context of two proportions, or the hazard ratio in the context of survival, and use that to recalculate like basically recreate what the updated P1, P2 values or the updated lambda 1, lambda 2 values are in these cases. Now, there is technically some more complex approaches to survival, but the most common one you'll see that is kind of calculate the overall exponential hazard rate and then use that to basically recreate, given some hazard ratio, what we believe the updated hazard rates to be. And I've actually given the formula here for the conversion for the two proportions case. And obviously this would apply for the chi-squared test, exact test for the two sample case for proportions, and the log rank test, Cox regression test for two survival. And then for two counts, we're talking here about, Poisson regression or quasi-Poisson or negative binomial, etc. And there's kind of two levels to this. There's the overall event rate, which is basically recalculating the per group rates, the count rates, basically using the same process as for two proportion to survival. But we also have this additional parameter if we're using quasi-Poisson or negative binomial Anderson-Gill, where there typically tends to be an additional parameter there to model what we call over-dispersion. And that over-dispersion concept is based on the fact that for the Poisson distribution, we expect the mean and the variance to be the same. And so over-dispersion measures how much higher the variance is above the mean for a quasi-Poisson model. And there's ways that you could kind of get estimates of dispersion for negative binomial from over-dispersion and vice versa, and similar for the Anderson-Gill. Given some assumption for Anderson-Gill, it's a slightly different method. But effectively, that means over dispersion, that concept is more like the variance. It's an independent variable. And so in this case, maybe you don't want to do the overall rate. You could just want to recalculate the over dispersion and use a similar strategy as the two means case. But if you're doing the overall event rate and updating the per group count rates, that's more similar to two proportions to survival. Or you can do a combination of them where you do both simultaneously. And then, there's a variety of other parameters that might be of interest. So there's R squared for ANCOVA, ITC for mixed models, CV for crossover, prevalence for sensitivity, specificity, discordant pairs for pair proportions, correlation if you're talking about co-primary endpoints. There's continuous monitoring approaches, there's Bayesian approaches, more and more and more. But, we don't really have too much time left here today. So what I'm going to do is kind of illustrate for you, hopefully quite quickly using simulated data that, the blinded SSR approach, the internal pilot approach, the value it has compared to the sample size calculation we did just there a moment ago. And so if we go to this table here, blinded SSR, I've done a little bit of prep work here, just like these values are more or less the same as what we saw, in a lot of calculations up to this point. And in effect, we're going to assume, in this case, we're actually going to use the original standard deviation assumption of 1, which obviously gives this sample size of 527 per group, or obviously 1054 if we double that for the total sample size. And so what I'm going to do here is actually recreate the pilot sizes that we got here in this table when we were trying to minimize the total study size. So obviously we're cheating a little bit here because we're kind of deriving these from a separate procedure, but let's assume that these were the correct pilot sizes here. And what you'll see here is that this table for an internal pilot approach is basically, now we're saying for the first 91 people of the planned 1,000 or so, so roughly the first 10%, we're going to do an internal pilot. We're then going to recalculate the standard deviation, and then we're going to use that updated estimate to then recalculate the total sample size. But that initial 91 people, that's going to be included in our final analysis. That's not getting thrown away. That's going to be included. And of course, you would hopefully expect that that will reduce the overall sample size expected. So what I've done here is I've simulated some data for these 4 scenarios. So, you know, I have 91 here. So I've basically simulated basically 2 normal distributions with one having a mean of 0, one having a mean of 0.2, and both having a standard deviation of 1. And basically what I'm going to do here is I'm going to take these values and I'm going to copy and paste them into this helper side table here. So rather than just like, you know, putting this in directly, this tool down here, the side table, allows you to calculate that based on actual internal pilot data. Or in theory, you could do this for actual pilot data if you wanted to use this tool compared to the other tables I showed earlier. Sorry, just copy and paste can be a bit fiddly. Yep, there you go. So we copy and paste that in. And so you can see here, you know, for the one sample variance approach, I'm going to just show this for illustration purposes. You know, the internal pilot, you know, the actual standard deviation, the overall standard deviation is just above 1. And so, you know, the sample size is very minimally affected. But what if we wanted to do the upper CI approach? And what if we wanted to make that comparable to what we did in our example from the first example? And so you can see here that the upper limit is 1.0727. That's very close to what we got from here. It's obviously a little, you know, given simulation randomness, there's a little bit of a difference there. But you can see that therefore now the total sample size increases to 1212. And you can see that's quite comparable to the sample size we got here, or the sample size that we saw here of 1205. But the main thing here is that that means that the total sample size for this simulated scenario is 1212, which obviously is, you know, around 100 less than the total sample size if we did an external pilot and then a main study. So obviously you've made that big saving of 100 people compared to that with the exact same scenario and the exact same assumptions about recalculating the standard deviation based on the internal pilot rather than external pilot. And of course, if we were to recreate this for the other scenario, for the 95% UCL, obviously we're doing 44 in this case, so we're doing, you know, half from each group as before. You know, we select this here, we'll paste this in, and you can see, you know, similarly here again, you know, the sample variance would give back more or less the same standard sample size that we have here. Initially, but obviously when we change this to be the upper confidence limit approach, you know, now it's increased to 1282. So obviously, if we compare that to the original example, you know, 1293, once again, random variation from the simulation, but these are very comparable. But of course, once again, the total sample size for this program, where we had an external pilot, is around 1400. Now, this is much closer to just what this initial main study sample size would have been in the first place, because we're getting to include the pilot data directly into the estimate. And of course, if we do the final one, there's no real comparison to the NCT approach for an internal pilot, but we can effectively simulate this as well. We'll just use the overall sample size for completion. Now, what I was hoping to do was to go through more detail on kind of a hypothetical example. That was the second example in the slide, but I'm not going to be able to do that in much detail today, but I will briefly go through it just for illustration purposes. So if we go back to our slide here, you know, what if the true standard deviation was 1.25 instead of 1? So effectively, you know, we have the exact same situation here, where we calculate it based on 1054. But what if the true standard deviation was actually, you know, 1.25? And what I've done here is I've just created a pilot study set where it's equal to 1.25. And then I'm going to input that here. And basically, this is probably a more standard scenario where, you know, we underestimated the variance in your initial calculation, and now the sample size has to increase because, you know, if we'd known that the actual standard deviation 1.25, then this would have been the sample size we would have had in the first sample size. But because we've done an internal pilot, we've now ensured that the study isn't underpowered because we made a mistake in our initial estimate for that parameter. And just a note, the code used to generate those files, and those files will be included in the save files. There'll be a link to the templates that are provided alongside this. So if you're interested in those files, you can, and to replicate it at home, then those will be available to do afterwards. Those will be alongside the slides and the recording. I'm going to very briefly do this two proportions example. In effect, the two proportions, what we're going to do is we're going to calculate the overall proportion from a pilot size of 20. Now, in this case, because, the pilot calculation is trivial, we're not going to have a side table like there again. Effectively, all we're going to do is we're going to specify the size of the pilot and then how many successes we got, or the proportion that we got. So 0.9, 0.6, and then a power of 80% with a sample size ratio of 1 gives a sample size of 63. Now, that if you round that up to the nearest even number, that will give you back the sample size expected of 32 per group. Obviously, that would give 64, but this minimizes the total sample size rather than the sample size given, an even number. Obviously, we expect an even number for sample size ratio. And we'll say the internal pilot size was 20, and then the internal pilot proportion was 0.7. Now, of course, that's lower than what we would expect given this assumption, where obviously the midpoint between 0.9 and 0.6 is 0.75. So if this had been true, we would expect this overall proportion to be 0.75. But we can see that because this moves slowly towards 0.5, the variance is maximized at a proportion of 0.5, the overall proportion is 0.5, just to be clear, under this assumption. So the more this moves towards 0.5, the higher the sample size is. And you can see there's a small adjustment here where maybe the sample size should have been 71 or around 35 per group or 36 per group rather than 32 per group. And so that concept is relatively simple to do. And then very briefly at the time available, it's not in the slides, but actually I was just simulating this earlier today, is basically we can also apply this to the count model in nQuery. The one we don't cover at the moment that I mentioned earlier is to survival, but that is in our development pathway for the software in the future. So if you're interested in that, certainly let us know and we can prioritize it above other projects that are in our development pathway. And so what I have here is basically, I have two examples here where I believe this is one. Basically, I had a Poisson process where the rate in group 1 was 1, the other one was 1.5. And in effect, what we were interested in is effectively kind of seeing, okay, what would be the effect of that of over dispersion in that case. So let's assume initially the sample size estimate was for an over dispersion of 1. So there was no over dispersion, the mean and the variance are the same, the kind of standard Poisson assumption. And then for a power of 80%, sample size ratio 1, we have a sample size requirement of 160 per group. So that's the number of people per group. But if we go to internal pilot size and we set that to 80, so let's assume that was like half the cohort. So we're going to have half the cohort, and that might be half the follow-up time. So in a count scenario, it's really time is the main unit, like basically the total time is the number of unit person years, as they're often called, or person weeks or something like that, is the real sample size. But that's just obviously the product of the number of subjects times the amount of time that followed up for. So when I put in 80 here, when I put 80 in here again for the total follow-up time, this is for 80 people, so we're gonna have 80 results. But if I put 80 here, that's basically saying I have the complete follow-up for these first 80 subjects, and now we're gonna recruit the next 80 people. But that could also equally be that we followed everyone perhaps for half the time if we were to set the sample size at 160. So that is possible as well. So you could set this to 160, set this to 80, and that would be equivalent to these events being for half of everyone's follow-up. And I've simulated some data here. So the first thing I'm going to do is going to give you some simulated data that didn't have dispersion. So this is basically assuming that the dispersion was 1. So there was no over dispersion. Or so the over dispersion was 1, so there was no over dispersion. So we paste that in. We see the sample size is pretty much equivalent. The estimated over dispersion is nearly 1. The internal pilot event rate is 1.2, which is quite close to the 1.25 we'd expect if we average these two together. And so overall, the sample size comes out as being pretty similar, right? But if I was to replace those instead with the dispersed value, and these data was simulated under a Poisson model where the rate in each group was 1 and 1.5, and the only difference between these two simulated data sets is that we created an over dispersion, a quasi-Poisson model, an over dispersion in the second data set. So the first data set, it was simulated from Poisson. This is simulated from quasi-Poisson or over dispersed Poisson. And if I input these values over this instead, we recalculate it, we see that the over dispersion is 2. So that's the actual value, the true value that I simulated from for the quasi-Poisson distribution. And you can see that the overall event rate is still roughly in the right area of about 1.25. And you can see now that the sample size is roughly double. And there's basically a direct relationship for the variance. Basically the over dispersed variance, if we're kind of using normal approximation, is basically equal to the variance times the over dispersion. And if you know your sample size formula, that basically means there's a direct proportionality between over dispersion and the sample size. So if you double the dispersion, you will double your sample size requirements for a Poisson problem when moving from Poisson to quasi-Poisson. So I don't have time to cover that in detail, but if you're interested in more, I'm happy to answer questions about that via email and go into that in more detail. So I think we're just out of time, so I'm not going to have time to answer questions. But just in conclusion, pilot studies are smaller studies undertaken to better understand the feasibility and likely operating characteristics of future primary study. For example, you might want estimates for sample size determination. Many pilot studies have poor characteristics. There's a really good review papers by Julius's group, who I've obviously referenced many times, for pilot studies in the context of UK and databases there. And the variety of different pilot studies is great, and a lot of them have questionable choices. But I would say, as a rule of thumb, if you don't have at least 20 people and you don't have decent sampling and decent population estimate, you're probably in trouble. So, pilot studies, if they're not providing you good information, then they're either, research waste because, it's not of much value for your main study, or are actively harmful because it might lead you to making over-optimistic assumptions about, say, the sample size for your main study. Inadequate sample size is one of the many reasons why your pilot study may be inadequately designed. Reasonable heuristics exist, sample size formulas exist. I would certainly encourage you to take them. And then just a final note that, external pilots have a lot of issues, but why not consider an internal pilot where you just take the first cohort of your study, the first 20%, first 30%, 40%, something like that, and treat that as if that was an external pilot, update your sample size the same way, but now you get to keep that data in your final analysis for free, and oftentimes be able to do that without any adjustment, basically treating that data as the increased sample size if that was the original sample size in the first place, and not worrying about having to adjust your t-test or chi-square test, etc. So that's the conclusion. Apologies for running a little bit over. As I said, I don't really have time to answer any questions, but do put your questions in if you have them right now, and I'll answer via email later today or early tomorrow. Just a note, as I mentioned here, nQuery is available, of course, to purchase, but obviously it's a tiered product. So either if you don't have nQuery and want to try it, or you want to try the higher tiers of nQuery and you don't have available in your current license, go to statsalts.com/nQuery-demo. And basically, your email has to be brought in here, and then that email will then send you a link to try nQuery within your browser for 14 days. No credit card details, nothing like that. Just literally try it and see if you like it. There's a little bit of a performance lag due to the online nature of the demo, but more than made up for the convenience of this, I would say. And just finally, if you have any questions that you don't have time to put in here today, or you think of later, go to info@statsalts.com, and that question will get to me. I'll be happy to answer that on behalf of our support team. And if you want to know more about StatSalts and nQuery, you can go to our website, statsalts.com. There's a wide variety of resources there available, including our entire webinar backlog from the last 3 or 4 years. It includes many templates, which effectively include save files and small examples including many from the webinars. They have written up examples of sample size calculations, which basically we replicated them from the literature from the New England Journal of Medicine and other resources. So lots of really useful information there if you want to know more. And as mentioned, the references for the 3 sections of this webinar are provided at the end of the slide deck. So if you want to know more, these resources are a really good place to start, and they're roughly ordered, I'd say, in terms of importance and in terms of separate areas. So without any further ado, I want to thank you so much for taking the time to watch today's webinar. I hope it was a really interesting part for you. This is obviously a relatively unexplored area for sample size determination, I would say. So hopefully you have some really good insights into how to design better pilot studies in the future, and the thoughts and ideas and context that you might need to appropriately size that pilot study, and maybe also, the idea of doing internal pilots, which kind of piggybacks off the external pilot literature and provides maybe a better mechanism via which that you get the advantages of a pilot data, but without the disadvantage of having to throw away the data. So hopefully some useful insights there. Hopefully you enjoyed the day. Our next webinar will be presented by my colleague Dennis Desmond, who will be doing it on non-unity hypothesis types, so non-inferiority, superiority by a margin, equivalence type hypothesis, and I think it'll be a really interesting topic. So if you're interested in that, make sure to sign up as soon as that becomes available on our website, or sign up to be automatically included in our emails for our webinar series. So without further ado, I want to thank you again so much for taking the time with me today. I hope you have a really great day, and I'll talk to you again soon.